All right, so our first step for that FASTQ file, remember, is alignment. So we now have our RNA-seq reads, and we will generally align our reads to the reference genome. The bulk of our reads are going to map to known exons and genes, which is here. So a small but sizable subpopulation are going to map to the genome, but not overlap any annotated region at all. And so these could possibly be, something like a novel exon or a novel gene that we haven't seen before. And then still others could be, unmapped reads that, we might have to investigate a bit further. And so remember that, in the creation of mRNA molecules, we splice in and out certain introns and include certain exons. And so there's a lot of isoforms of specific proteins that change the function of that protein quite drastically. And so these unmapped reads could possibly be novel splice junctions that we haven't seen. So what we have kind of aligned to our reference and quantified in these experiments is a random sample of cDNA fragments derived from RNA in our sample. And basically, we're relying on the assumption that probabilistically a gene with increased expression in a given cell is represented by more NGS reads, so more of those short sequencing reads. And basically, after we map those to the reference genome, we have a matrix or a big giant table of rows and columns that correspond to our experimental information. And so usually we have the genes in rows and our samples in our columns. And so you can see we have 2 wild type samples here and 2 mutant samples. And we can start to see, the RNA-seq counts that are there for each particular gene of interest. And basically what we're doing is we are comparing, our groups of samples. And so we have 2 of wild type and 2 of mutation. And so we compare expression in, say, gene 24 of wild type versus the 2 mutants. And we do this for, tens of thousands, about 20,000 genes in the human genome. But our goal is to compare the gene expression, in our biological replicates. Now, there's a question there at the bottom that I want you to sort of think on, before we move on to the next slide. Now, what do you think would artificially create differential gene expression between samples when we are comparing them? So not biological differences, artificial differences. So things that are inherent, in the transcripts themselves. And if you had an idea, maybe longer genes, would tend to have more reads mapped to them, since we're using this concept of these short sequencing reads to quantify how much of a gene is expressed in a given cell. Maybe the really, really long transcripts would just, by virtue of being very long in the genome, have more RNA sequencing reads. And you'd be absolutely correct. And then if we had very different sequencing depth between our samples, and so, for example, let's say that in our previous example, the 2 wild type samples were sequenced at very low coverage. And so we didn't have a lot of sequencing reads. And then the 2 mutant samples were sequenced at extremely high coverage. Well, then we'd basically see that every gene in our mutant sample was upregulated compared to our wild type. And this isn't actually true. It's artificially created based on the parameters that we gave the sequencing instrument. And so what we need to do is this step called normalization, or to normalize between our samples. And we're not going to get too much into kind of the mathematics behind normalization, but basically what we need to do is we need to correct for those things. So we need to correct for the fact that longer transcripts tend to generate more reads compared to shorter transcripts present at the same abundance in the sample. And then also the variability in the read depth produced from each run tends to fluctuate. And so we need to correct for these to be able to make a valid comparison between our groups of interest. And basically what we get is something like this. We get a table. You see that our read counts have now been normalized, and so there's been some sort of adjustment that allows us to compare between our specific groups. You see our genes in our columns, or in our rows here. And then you see other metrics that are generated by doing this differential expression type work. You can see mean expression of any particular gene. A big criteria that's used in RNA sequencing studies is this idea of the log2 fold change between your groups. And so basically, this is a measure of how much, or what's the best way to put this? It's a measure of the degree of change between your specific groups of a given gene. And so another way to say this is, on a log scale. So CMIP, in, let's say, this EMP208 group is, this much more expressed than your group that you're comparing it to. So say we're comparing it to or let's say we're comparing num20 to EMP-docs. And so on a log scale, num20 is increased in expression. And so this is just a measure of the magnitude of the gene expression change. And then we also do some statistical significance testing. And again, the p-value is often used as kind of a significance cutoff that allows us to kind of get a short list of genes that give us sort of a gene expression signature of our particular group of interest. And so I know that was a fairly quick explanation of RNA-seq, and I think RNA-seq is really well understood if we actually get in and actually do some analysis ourselves. And so I've also uploaded a PowerPoint called Exploring Gene Expression with R. And so we're going to return to our very old friend R. So I know a few of you emailed me and said that you would like some more experience with R since it is free and available, and you can use it for data analysis. And so here's our shot. So I think going through the PowerPoint, I've made it pretty easy to follow along. Try your best and see if you can do it. If you give it a shot over the weekend and you're really not getting it, I'll actually upload a video of me doing it within R on Monday, and then I'll hold some live office hours on Tuesday for any questions related to this material. And so give it a shot first, see if you can get it. If you can't, then you can check out my video on Monday. All right, thanks guys. Have a nice weekend.